Hello students. In our previous lecture we have already studied about the two laws of Mendel, which was law of dominance and law of segregation. Now here we are going to study the third law of Mendel, which is law of independent assortment. We have studied that both the other laws, dominance and segregation, was based on the monohybrid cross, but this one is based on the dihybrid cross. Now let us see what is this dihybrid cross first. See, dihybrid cross means two different plants getting crossed. Okay, two different characters are crossed in one single plant. So for this experiment, Mendel has chosen two different plants to make one, and then other side he has crossed with the plant which was having two different traits and two different characters. Let us see that with example. Mendel has chosen actually a round shaped seed which was having yellow color, and this trait he has fused in one single plant to get crossed with the other plant which was having wrinkled seed, and this seed was green in color. So this is called dihybrid cross, where two different traits are fused in one single plant and then they are getting crossed. Okay, so let us make the gametes of them first. For round and yellow, actually they both are going to be always dominating. They have always shown the dominating nature, so they are represented or expressed with the capital RR and capital YY. Same way here, these both are going to be always recessive, so they have this kind of a alleles. Now when they will be getting crossed, obviously they are going to be from diploid to the haploid, from to end to the end. So the gametes should be there. Now let us make the gamete. You know, at the time of a gamete formation, out of two genes, out of two alleles, one is going to be come in the gamete. So from here one R will come and from here one Y will come, right? Same way here R and Y. This is going to come in the gamete. Okay, so now when these both the gametes will be fused and they are going to make the zygote, they are going to get join over here. So capital R, small r, capital Y and small y will be expressed here. But in this form, when Mendel has seen his first progeny F1, that is phylum 1, he found that all these plants are round and yellow. So again he has to see, he has to check the genetic character also. So again on the basis of this result of F1, he went for the F2. And in F2 again he had done selfing of these progenies only. So when again he started making gametes, he had made 4 different gametes, and these gametes were capital R with capital Y, capital R with small y, and here small r with capital Y and small r with small y. These were the 4 gametes which he have found from this. Okay, same way if you will express it, if you will expand it, same way like this you can expand and you can see the same result is going to come. Okay, so in F2 generation he made gametes like this, and when he made gametes like this, he found that almost all the characters are expressing itself in this experiment in the F2 generation, because F1 all were round and yellow, and F2, because of these gametes segregation, he found that all the progenies are different. There are many various characters which are shown, and all these 4 are exhibiting themselves in one or the other offspring. So in this he had got a phenotypic ratio, and in this phenotypic ratio was 9:3:3:1. This was his phenotypic ratio, and this is the example of your dihybrid cross where Mendel has proved that each and every character is expressing itself in the F2 generation. So hope this is clear to you. If you want, you can copy it down. Now after this, I hope you are clear with both the laws of all the three laws of Mendel. Now again here one question arises, and that question is, how are you going to know that whether the plant is homozygous or heterozygous by looking it? For example, you can see this. If it's a pea plant, okay, so if you're watching this pea plant, you can see that this pea plant is tall in nature, right? So now, as if this plant is tall, can you guess that whether it is homozygous or heterozygous? No, na? Just phenotypically you can say, oh yes, it is a tall plant, right? But for tall pea plant, there are the two cases which can be. Either this can be pure tall, or it can be like this also. This is homozygous and this is heterozygous, right? So in this case, there are two different alleles, right? One which is showing tall nature, dominating one, and other one is recessive, right, which is dwarf. But my plant is tall, so I'm not going to see this recessive character. I have to find out whether my plant is homozygous or heterozygous. So to observe this kind of a plant, we do a testing, and this testing is called as a test cross. Okay, so we have seen here two different cases. This one is case 1 and this one is case 2, right? So let us see and let us prove ourselves that how it is going to be the correct one and which kind of a case is in my plant, right? So let us see both the cases. This is case 1 and let's make here case 2. So now let us see our both the cases with the making a Punnett square. Let's make a Punnett square here. Okay. Same way here also we are going to make a Punnett square. Now let us fill it. Here we are going to take this chance, second case, capital T, small t and small t with the small t. Okay, so let us see this. Let's make a change. Okay, so let us see this. What will happen over here? This is capital T. Here small t will come. Here capital T, small t will come. Here small t, and here small t will come. Right? So in this case, there is a 50-50% chance, right? So 50% is going to be tall, and here 50% is going to be the dwarf plant, right? Let's see our second chance, second case. In this second case, let us take pure tall plant with the pure dwarf plant. Okay. Again here, this kind of gametes are going to get formed. So in this, what you see, in this case, all the plants are going to be tall. So here 100% chances of tall plants are there, right? So here in this plant, I can say that it is heterozygous. It can be heterozygous because 50% chances are here which are showing tall character, and here all are heterozygous. 100% plants are heterozygous, right? So in both the cases it is there that this particular plant is a heterozygous. Did you get this? Let's see once again. Here this is dwarf. This is dwarf. Tall is only these two, and these two are not homozygous. This is homozygous. This is homozygous because both are the same, and here both are different, right? So as they are different, they will be called as a heterozygous. So 50% chances are here. So wherever in exam if question is asked that which cross shows the 50% ratio, you are going to say it is always a test cross, and test cross is made to identify whether the plant or whether the organism is homozygous or the heterozygous. Okay, so this is all about your third law of Mendel, that is your law of independent assortment and the test cross.